Welcome to our live streaming guests as well. So this is the third general session at NITOP, and before I introduce our speaker, I wanted to just have a few announcements. So today at lunch, right afterwards, we have NITOP Nearly Impossible Trivia of Psychology. NITOP. At this activity, you sit with a table of people, and they become your team. You'll have 10 minutes to answer 8 nearly impossible trivia questions, and the winning table gets free drink tickets, which leads me to the next announcement, which is tonight is NITOP karaoke night at 8 p.m. In the Jacaranda Hall. This is not just regular karaoke. This is guilt-free, shame-free, privacy-insured karaoke. So please feel free to come by and check that out. It's fun for everyone. Also, today we'll be sending you a link for the conference evaluation that will be coming through email, not through the app, because we're sending everyone a unique link, so that way you can complete it as you go. You don't have to do the whole thing at once. So please check your email for that link for the evaluation. We take those very seriously. Tomorrow at the end of the conference, we'll drag ourselves up to a meeting room, and we will start planning next year's conference. So it's my pleasure to introduce our general session speaker today, Dr. Elizabeth Phelps. Dr. Phelps is the Pershing Square Professor of Human Neuroscience at Harvard University. Dr. Phelps earned her PhD from Princeton University in Cognition and Cognitive Neuroscience. Dr. Phelps has had an outstanding career in both research and teaching. Among her many awards, she most recently received the George Miller Prize from the Cognitive Neuroscience Society in 2021 and the William James Award from the Association for Psychological Science. She's a fellow of the American Association for the Advancement of Science. Dr. Phelps has served as president of multiple academic associations, including the Association for Psychological Science. Dr. Phelps' talk today is sponsored by W.W. Norton, and we deeply appreciate their support of NITOP. Today, she will address us on how the cognitive neuroscience of learning can inform teaching of psychology. Please welcome Dr. Phelps.
So much. So I just want to start by saying I am delighted to be here. I teach psychology, and I've taught intros to psychology for decades, longer than I want to admit. But I'm rarely in a room of other people who are equally interested and excited about how to improve our teaching of psychology. And so thank you for having me. I'm going to talk today. So I'm a cognitive neuroscientist, which means, of course, I study the relationship between the brain and behavior in humans. And this is something that I do research on, but it's also something that I think about as I teach psychology. And so I'm going to just review for you today a few of the things I think about from my research that informs how I might structure a class in teaching Introduction to Psychology, or any psychology class for that matter. And so one of the goals that we have when we teach psychology is to create a learning environment that enhances the retention of insights from psychological science. And I think about 2 areas of research that I conduct that specifically might inform this goal. One is enhancing memory retention through reconsolidation. So I'm going to spend a lot of time today sort of unpacking what I mean by reconsolidation and what the data is behind reconsolidation. And the other is enhancing memory retention with mild arousal. So before I do that, I want to give you, pushing the wrong button here, some background on the neurobiology of memory. So this may be familiar to many of you, but I just want to make sure that we're all on the same page before I start diving in to the brain science a little bit more. And specifically, I want to make a couple points. We know that there are multiple types of memory, and they have different means of expression. And these different means of expression and these different types of memory are dependent on distinct neural systems. But of course, in any situation, all of these memory systems are active, and so they can also interact in specific situations. And today I'm going to focus on 2 memory systems. The one that we think about the most, which we call declarative or explicit memory. This involves the conscious recollection of events, that's episodic memory, or facts, semantic memory. And then you know that this depends on the hippocampus and parahippocampal regions, regions sort of surrounding the hippocampus, for initial storage, whereas additional brain regions will come into play when you have to retrieve information. The other memory system I'm going to talk about, which is specifically related to emotion, is an amygdala-based memory system. This underlies threat memories, and it involves the expression of learned defensive threat responses. So not do you tell me what you remember, but is your body showing me what you remember through freezing, your actions, or perhaps your physiological response like autonomic arousal. And we know that this depends on the amygdala for both storage and retrieval. So just to come back to a little bit more to explicit memory, so we think of this as a primary memory system in humans. When we think about what is memory, we think about our ability to consciously recollect facts and events. And this is the memory system we care about the most if we want to help our students retain the information that we teach in the classroom. We know that the medial temporal lobe, including the hippocampus, is important in this memory system. And here I'm sure all are familiar with the famous case of H.M., who we now know, now that he's passed away. His real name is Henry Meliasen. That was kept secret for years and years and years. H.M. in his early adult life had surgical resection of the medial temporal lobe. This included the hippocampus and other cortices. We call them parahippocampal cortices. And we know that he, as a result, had a hard time remembering essentially anything in his life. I was privileged enough to meet H.M. once. It was really fascinating. He really, from one moment to the next, didn't really know what was going on. And in that way, he was in some ways the perfect experimental subject, because he didn't know he'd been there being tested all day. At the same time, it kind of made you feel bad about testing him all day. But he really, from one moment to the next, didn't kind of know what was happening. Although he could engage in a conversation, he had very good short-term memory. The hippocampus here in blue, is a small structure in the medial temporal lobe. Gets its name from the word for seahorse. It sort of has that shape. And it's been identified within all the regions that were damaged in HM as the one that's most critical for the storage of memory. Although semantic memory, the ability to learn new facts, is also dependent on regions sort of right outside the hippocampus. The threat memories involve the amygdala. This is not critical for the learning and retention of psychological science, but it does inform our understanding of reconsolidation. And we also know that the amygdala interacts with the hippocampus to facilitate explicit memories. That's why I'm focusing on this memory system today. So how do we study the amygdala? The typical paradigm we use is Pavlovian threat conditioning. So here, you'd have a participant who would come in. They would see a neutral stimulus, such as a blue square. It would be paired with a mild shock to the wrist, for instance, something aversive. It could also be something rewarding. And this mild shock to the wrist will result in an autonomic response, such as a skin conductance response, as you can see here, initially to the shock itself, but eventually to the blue square that predicts that you will see a shock. We know from lots and lots of research in humans and rodents that the amygdala, here in red, and what I've done on the right here is actually take an MRI scan of a participant and made their brain glass. Here's the front and here's the back, and trace an actual amygdala and the hippocampus. So the hippocampus and amygdala are right next to each other. And we know that the amygdala, from research in rodents, is necessary for the acquisition of this learning. The storage of this learning is in the amygdala, and it's also necessary, of course, for the retrieval of this type of learning, as expressed through your physiological response. In humans, hi, sorry. Should I take one off?
No.
I am really mic'd up here. Okay, in humans, we know the amygdala is important for the expression of this type of learning, because patients with amygdala damage fail to show this sort of increased physiological response to the stimulus that predicts that they're going to receive a shock. So here on the top, I've plotted the skin conductance response for a healthy participant. He shows a response to the shock itself. but also to a stimulus that predicts the shock. Whereas a patient
notice if there's anything odd.
Sorry, didn't mean to do that yet. Whereas a patient with amygdala damage will show a response to the shock itself, so it's not the case that their autonomic nervous system doesn't work, but they fail to show this association as expressed through an autonomic response. However, these patients, especially if they have an intact hippocampus, do have explicit knowledge for the events that occurred. So here's a woman with bilateral amygdala damage. This is the cartoon version of her data that you're seeing here. She was shown her actual data and asked to comment on it. So I'm going to show you what she said.
About my reactions to normal lifestyle, life threats. My fear element seems to be, to me, in my head, normal. I've never noticed anything odd.
And how do you feel seeing these results? How's it?
I'm dead. I think that it's probably absolutely correct. I know I don't sweat. I knew that there was an anticipation that the blue square was going to, at some particular point in time, bring on one of the volt shocks. But even though I knew that, and I knew that from the very beginning, except for the very first one where I was surprised, I really don't think that I mean, I knew it was going to happen, but that was my response. I knew it was going to happen. I accepted that it was going to happen. So I learned from the very beginning, it's going to happen. Blue shock. Happened. I turned out to be right. It happened.
So I think she dramatically demonstrates, even though she fails to show me evidence of this learning through this physiological response, which we know is amygdala dependent, she has very good knowledge of the events that occurred, which is, of course, hippocampal dependent. And if you take patients with damage to the hippocampus whose amygdala is intact, you'll show the opposite pattern, which is they will show this physiological response to the stimulus that predicts the shock, but they'll fail to be able to tell you that the blue square predicts the shock later on. So just to summarize what I've mentioned already, we know that declarative or explicit memory involves the conscious recollection of events or facts, depends on the hippocampal regions, and is the primary memory system needed for the retention of information taught in psychology. Threat memory involves the expression of threat responses, depends on the amygdala, and can inform our understanding of reconsolidation, which I'll get to next, and can interact with, but is not critical for explicit memory retention. So I want to now move to memory reconsolidation. Reconsolidation is a topic that has been investigated quite a bit in the last 20 years or so. So I want to start with just saying, what is it? And then I'm going to highlight, what's the early evidence for reconsolidation, and then why was it so exciting? Why did people get so interested in this? There are now thousands of articles on reconsolidation, primarily in rodent work, but a good, few hundred at least in humans as well. So what's the initial evidence in rodents? What was the initial evidence in humans supporting reconsolidation as a memory mechanism? What induces reconsolidation? So when can you trigger reconsolidation and see some benefit of this perhaps? And then how can this inform teaching psychology? So first, what is reconsolidation? Until about 20 years ago, if you asked any neurobiologist who studied memory, they would give you sort of this standard view of memory. And the standard view was that you learn something. There's a period of time where the memory is consolidated. During this consolidation process, the synaptic changes that form the memory occur. This requires protein synthesis. So if you were to, right after you learn something, inject something into the brain that blocks protein synthesis, you would block the storage and consolidation of that memory. In humans, we probably see this most often in something like when someone has a concussion, they may not remember what happened right beforehand. We think that it's because they blocked the consolidation of that memory, the concussion did, or ECT, for instance. But it was traditionally thought that once the memory is stored, and it's gone through the full consolidation process, it's there. And you can retrieve that memory by sort of pulling out that memory and then putting it back wherever its storage was. And then a second time, you retrieve the same memory. Like you take a file out, put it back, take a file out, put it back again. This was the standard view of memory until about 20 years ago. But there was an alternative view of memory, really first talked about in the 70s, but not really verified with data until the 2000s, in that actually there's multiple consolidation processes. So here the idea is you learn something, there's a time-dependent process where plastic you know what I'm saying. I'm so tired, I flew in late last night. Where the memory's formed. There's plasticity in the brain. Once the memory is stored, however, when you retrieve their memory, some of the times, there is another period of synaptic plasticity. And what this means is even after a memory has been initially stored, if you can disrupt that second process of synaptic plasticity, you might be able to change the memory or disrupt the memory. And so the notion was that with retrieval, at least some of the time, we're going to engage in a second storage process. So what's the data supporting this? This is Kareem Nader. He's now a professor at McGill University. At the time, he was at NYU, where I was. And he did studies in rodents using a simple threat conditioning paradigm, a Pavlovian threat conditioning paradigm. So much like the one I showed you earlier, he took a rodent. He played, in this case, a tone. He paired the tone with a foot shock. The rodent started to freeze then when it saw the tone, or when it heard the tone, excuse me. And so this learning occurred on the first day. He waited a day for this tone shock memory to consolidate. He then brought the rodent back into the laboratory. He just played the tone by itself just once, sort of not a repeat of several tones like he did during learning, but a single tone presentation. He then injected into the lateral nucleus of the amygdala, where we know from work in rodents is a site of storage of this memory. He injected into the lateral nucleus of the amygdala a protein synthesis inhibitor, which should block the storage or restorage of the memory. He then waited a day. And I should say, if you tested right then, before the memory was restored, the rodent would still freeze. Okay? But if you waited a day and he played the tone, the rodent didn't freeze at all. It's like they never had the experience with the tone and the shock. So this was very exciting work, suggesting that even after a memory has been consolidated, you can disrupt its restorage or reconsolidation by injecting a protein synthesis inhibitor, and suggest a memory system that looks something more like this than the standard view of memory. Now, why was this so exciting? So those of us who study threat memories, this was a seminal finding, been cited thousands and thousands of times at this point. It was so exciting because this sort of threat memories have become a model for things like maladaptive threat responses, for instance, as you see in anxiety. And the standard way that we use to diminish threat responses in these learning paradigms is extinction learning, as described by Pavlov. So extinction learning, you have initial acquisition, a rodent hears the tone paired with a foot shock. They start to freeze more now when they hear the tone. Or in humans, they see a blue square, it's paired with a mild shock to the wrist. They start to show greater skin conductance when they see the blue square. But if you now present the tone or the blue square repeatedly without the shock itself, you see a diminished freezing in rodents and diminished skin conductance in humans, and this is just classic extinction learning as initially described by Pavlov. And this, of course, forms the basis for exposure therapy treatments for the treatment of anxiety disorders and PTSD. But one of the problems with extinction, and this again was also first described by Pavlov, is that extinction doesn't result in erasing or changing the initial memory, at least not very much. What it results in is a second memory trace that now competes for expression with the first memory trace. And as Pavlov first described, with salivation, has also been shown extensively in threat learning. We know this because under a number of circumstances, with the passage of time, you get sort of the shock at sort of independently, the animal's stressed. Under a number of circumstances, the threat memory reappears. So the memory still has to be there. And so this is sort of the case of poor extinction retrieval. And this matters at least because, in terms of using this type of technique in exposure therapy, for instance, because it suggests that these maladaptive threat responses that might have been ameliorated through exposure therapy can actually return. And this is thought to underlie the high relapse rate for things like exposure therapy and CBT. And so people were excited about reconsolidation very much initially because it was thought that maybe targeting reconsolidation could be a more effective technique to reduce maladaptive threat responses. I'm happy to discuss in the question period where that stands, but I will move forward with the lecture about memory and teaching right now. So I should say, another reason why reconsolidation was so exciting is because it wasn't a memory phenomenon that was unique to threat memories. It was also shown that hippocampal-dependent memories and motor memories undergo reconsolidation. So in another series of studies, some from, this is from Jacek Debiak in the lab of Christina Alberini just a couple years later, showed that they could block context memories by injecting protein synthesis inhibitors into the hippocampus after a previously consolidated memory was retrieved, and show now deficits in retrieving that context memory. So this was a sort of a general memory phenomenon, not unique to just threat memories. So what about in humans? So what evidence? So in rodents, what they've done is they've sort of shown that this is due to synaptic plasticity by disrupting synaptic plasticity by injecting drugs into specific neural systems. We can't do this in people, obviously. And so we have to find alternatives. And this led people to say, well, why would it be the case? Why would it be the case that some of the times when you retrieve a memory, it undergoes the second storage process? And it's been proposed there's two functions of reconsolidation. One is to strengthen the memory. If a memory undergoes one consolidation process, it can be strong, it may be weak. If it undergoes a second consolidation process, it's going to be stronger. If it undergoes a third consolidation process, it will be even stronger. So a primary function of memory, of reconsolidation, is thought to be strengthening of memory. A second proposed function of reconsolidation is to update the memory. So whatever it is that caused you to retrieve that memory in the first place, whatever situation that was, may have information in that moment that's valuable for the future use of that memory. And so you can now update the memory with new information available at the time of retrieval. And this is thought to perhaps underlie the dynamic nature of memory that we see in things like misinformation effects. So to find evidence for reconsolidation in humans, we want to take advantage of this updating function and to see if we could update a threat memory with new information about the value of the stimulus, the conditioned stimulus. And this was work that was done in my lab by Daniela Schiller, and a rodent study was done in Joe Ledoux's lab simultaneously by Marie Monfils. Daniela is now at Mount Sinai, and Marie is at UT Austin. And so the way this study worked, at least as one of the studies, we've done several versions of this study. The way this study worked was participants came into the laboratory. They saw two neutral stimuli, here an orange square and a black square, each of which on some subset of the trials was paired with a mild shock to the wrist. They also saw another stimulus, we call this CS minus negative, conditioned stimulus negative, that was never paired with shock. This is a baseline stimulus. So I'm calling one of these the CS, the reconsolidation conditioned stimulus, the CSR, and the other one, the extinction conditioned stimulus, the CSE. And that'll become clear in a second. The second day, participants came in. So we waited a day for this memory to consolidate. The second day, participants came in, and they were given a reminder to reactivate one of the threat memories, in this case, what I'm calling the reconsolidation conditioned stimulus. Then there was a 10-minute break. So this was sort of an out-of-context, unusual, what's going on here presentation that just, was just one presentation, not a series of trials by itself. There was a 10-minute break, and then participants underwent what would be, we're calling safety training, but it's really extinction learning. They saw all three stimuli repeatedly with no shock, no aversive events. So the only difference here between what I'm calling the reconsolidation conditioned stimulus and the extinction conditioned stimulus is that the reconsolidation condition stimulus is undergoing this safety learning during what we think is the reconsolidation window, during a time when the memory is being restored, whereas the extinction stimulus is undergoing standard extinction learning. Participants came back a day later. They got some sort of just sort of random shocks. This helps reinstate or induce the recovery of the threat memory, and then we did another extinction session. So this is the data that we got from this. What I've plotted here on the y-axis is this skin conductance response and here it's on the end of acquisition. So what you can see at the end of acquisition is participants acquired conditioned responses to both stimuli that were paired with shock, but they didn't show a skin conductance response to the stimulus that was never paired with shock. So it just showed Pavlovian learning. This is the end of extinction learning. So here what you can see is this sort of safety learning reduced the conditioned response to both stimuli. And here's the beginning of the third day. And what you see here is we see robust evidence of a return of the threat response to the stimulus that underwent standard extinction. This would be consistent with sort of this recovery of threat responses that you typically see during after extinction learning. But we see no recovery with the reconsolidation conditioned stimulus. And we then re-extinguished everybody. I put this here only because in another version of the study, we brought people back a year later. We were still able to reinstate the conditioned response in participants that underwent standard extinction a year later, but showed no evidence of recovery if they underwent what we called extinction learning during reconsolidation. And so this suggests that we can sort of rewrite in some ways the original threat memory as it is stored in the amygdala to incorporate this new information that's available after the retrieval of that memory when the memory is undergoing this vulnerable reconsolidation process. So in humans, there's also evidence that this is not unique to threat memories. So here I'm showing you a paradigm that was conducted by Almut Hopbach, who's at Lehigh University. And what she did in this study was she brought participants in, she put a bunch of objects in a basket. And then she had participants go through and name the objects, and they were supposed to remember them. She then brought participants back 2 days later. They either were reminded of the original learning experience by being in the same context, in the same room, in this case, with the same experimenter, or they were in a different room with a different experimenter. And here they were given a bunch of objects that were laid out on a table, and they were asked to memorize the objects. And then they came back 2 days later, and they had to recall what they learned the first day, not the second day. And what you can see here is her data showing the recall of items from the first day. And people were somewhat better, not significantly better, at recalling items the first day if they didn't have the reminder the second day. But the bigger effect is that information learned the second day often intruded on the first day learning. So now, when you're saying, tell me the items you learned the first day, if they had the reminder, they're much more likely to incorporate information that they learned the second day. So they're having sort of a source confusion in this case. And it only goes one way. If they ask them to recall the second list, they never show information intruding on the second list from the first day. So this suggests that this is, again, a hippocampal-dependent task. It's a conscious recall. So this type of learning extends to the hippocampus in humans. So that leaves the question, what induces reconsolidation? I've been showing you examples where we do the single retrieval, and that somehow induces reconsolidation. But we know, and this is very important, that not every retrieval induces reconsolidation. So if we go back to this example that I showed you, what's the most puzzling thing here, and maybe some of you notice this and we're thinking this in the back of your mind, is that the first trial of extinction learning is also a retrieval, right? Yet, if I do extinction learning, normally I don't see evidence for reconsolidation updating. And so there's been a lot of research trying to say, why is it sometimes when you retrieve something you induce reconsolidation, and other times when you retrieve something you don't? And what this has come to is this conclusion that only incomplete, out of context surprising memory cues induce reconsolidation. It's been suggested that you need some sort of prediction error, something to tell you there's sort of something new to learn here. I'm not getting exactly what I expected, right? And so therefore, I need to update or change or strengthen my memory. So you need a prediction error to induce reconsolidation. So, when you have a prediction error, an incomplete reminder of sorts, you induce a memory model that looks like the reconsolidation memory model I talked about earlier. When you have retrieval, you get another storage process. When you don't induce a prediction error, when you have sort of a repetition of what you might expect, you have a memory model that looks something like the sort of standard memory model that I talked about. The memory, you might have a new learning trial, but you don't update the old memory. It is not restored. It is not reconsolidated. And so here's a sort of a review article that was a slide from a review article just to kind of emphasize how this works. So what you see here, this is a review by Sinclair and Barantz, is participants learned pairs of images. And this is just sort of cartoon version of what's happening in the brain, the representation of both of these images. Now, if you just repeat that pair of images as you saw them the first time, you'll get another learning trial. But you won't induce reconsolidation. However, if you present, say, just the cue and not the pair, which forces them to sort of say, okay, what's missing here, and try to come up with something, try to retrieve something, then you will induce a reconsolidation process. So by doing this, by giving incomplete information, you're generating a prediction error. This prediction error induces memory reconsolidation. And then there can be 3 consequences of inducing reconsolidation. If you do something to disrupt the memory at that point, some type of manipulation that disrupts consolidation or reconsolidation, you will then disrupt the memory. If you present new information at that time, you can update the memory. If you don't present any new information at that time, and then it will just strengthen the memory, just have a second consolidation process. So there's 3 consequences, potential consequences of reconsolidation. Disruption, which generally in humans for episodic memories takes some type of physical disruption, updating, or strengthening. So how does this inform teaching of psychology? Well, I think this is captured very well in what we call the testing effect, which many of you are probably very familiar with. The testing effect suggests that retrieval practice leads to better retention and better subsequent test performance than reviewing the material itself, just reviewing the material. And so, retrieval practice is essentially an incomplete reminder. What is it you learned yesterday? Who was the person that first described classical conditioning? So all of these things cause the person to have some prediction error, some incomplete reminder, that then can induce a reconsolidation process. Whereas if you just restudy, you will have essentially, sort of a second learning trial, but not reconsolidating the initial learning. And this is essentially what they find in the testing effect. And so based on the principles of reconsolidation, how does this inform the teaching of psychology? A few things, some of which has already been documented in psychological research on the testing effect. We know that retrieval practice after initial consolidation will be more effective. So if you do the test, if you do a sort of a practice test immediately or in the same class session, it will have a minimal benefit. If you do it in the next class session, it'll have a bigger benefit. Retrieval cues that produce stronger prediction errors may be more effective. And so incomplete recall or cued recall tests may be more effective than multiple choice recognition, where you're actually showing them all of the options. And again, both of these findings have actually been demonstrated in some of the psychological literature. And that feedback soon after retrieval practice could help update incorrect initial learning. So it may be the case that the memory trace formed initially is not correct. And so if you give feedback very soon after the testing procedure, that can actually help update the incorrect initial learning. So just to summarize this part of the talk, I've talked about one way we may enhance memory retention through reconsolidation. that by inducing reconsolidation of previously consolidated memories, this creates an opportunity to strengthen memories of classroom materials, and this may underlie the benefit of testing and provides insight into how testing and retrieval practice may be most effective. I'm going to take questions on that before I move on to the next section. You can also ask questions later on about that. But if there's any burning questions, I'll get to them now. Yes? Is that okay if I take questions right now, Steve? Okay, yeah.
Thank you. Could you just comment on how the time gap that you would need to maximize this effect?
Well, so I will say that we don't know the time gap that you need to maximize the effect, but a longer time gap is going to be better. So you can think of it as, a neurobiologist I know, Christina Alberini, describes it like this. You have initial learning, and then you have consolidation. Consolidation is strongest initially, and it decreases over time. It goes on for a very long time in humans. We know this from research on retrograde amnesia. But every time you then have this sort of this retrieval that induces a prediction error, you reboost it, right? And so it's almost like, so presumably if it's a little later, you'll reboost it even more, but it doesn't mean that there's not a benefit at any part in that sort of downward curve. But there's no sort of, one of the problems in this work is that a lot of the research is in rodents, and the timeline of reconsolidation changes a lot in rodents, and it hasn't been sort of investigated in precisely in humans.
Is it minutes, hours, days?
Oh, I'm thinking more definitely hours or days. Minutes, I don't think will do anything. So we know the reconsolidation process. So definitely hours at the minimum. But I'm sort of, days is better. Yeah, if that helps. Yes. And I should say, if you do a test initially, one of the things that the Kareem Nader showed, and it's been shown in humans as well, when you disrupt reconsolidation, like with a protein synthesis inhibitor, immediate recall doesn't require protein synthesis. You don't actually see a deficit immediately, right? Because the consolidation is a time-dependent process that goes on for a while. And so is reconsolidation. The time window for reconsolidation is shorter than consolidation, but still it goes on for a while. Yes?
Wow, that's a little bit louder than I expected. Two questions. One is a clarification on the study that you're talking about with the humans, with the extinction and the effect. Was that a between subjects or was that within?
That one was within. We've done it between as well.
Okay.
Yeah.
I'll have to go back and read it and understand exactly what happened. But
so I can just so everybody learned in the same session that 2 stimuli predicted a shock, and only one of them was reactivated.
Okay.
Whereas we've done it between subjects where they learned about one stimulus and it was either reactivated or not.
Okay. So okay, got it. And then the second question was when the reconsolidation process happens, do you think, and you said it's because there's a prediction error and they need to add new information, or they do add new information. Do you think the reason that they strengthen that memory or have better access to it is because they've created more access nodes or more pathways to be able to retrieve that information? I guess I'm going on to retrieval at this point.
So the thought, at least from the reconsolidation literature, is it really, memories are stored very weakly or strongly, and so this increases just the strength. It's not that it changes. It can, with updating information, change the sort of nature of the memory, but if nothing else interferes or comes in after, it just is thought to strengthen the initial memory, not change the initial memory.
Okay, so the synaptogenesis or the
great.
Thank you.
And I should say that the reason why we did that within subject version was we also did it in a scanner, and we were able to show in the scanner that that sort of the pathways you see involved in extinction aren't there when you have now updated the memory. Through reconsolidation. So it sort of confirms the idea that we're updating the memory as opposed to inhibiting the memory. Yeah.
Okay.
I have a question from our remote audience.
Yeah.
Should we add the threat memory in a cognitive memory model to explain the importance of social issues like sense of belonging, fear between peers, etc.?
That's a hard, I do talk about threat memories in terms of learning about others, right? And learning from others. So I talk about it more in terms of implicit bias at times. But I do think, this is just a basic learning mechanism. We learn from association, and that can happen in the social context. It can happen in a non-social context. When it's a negative thing, something you find threatening, I do think that can play out in social domains. But it's hard for me to answer that specific question. Should I move on? I'm going to move on at this point. Yeah. I just want to make sure I get through all this. So the second thing I want to talk about is how does arousal influence memory, and what does the neurobiology of arousal tell us about how arousal influences memory, and how can this inform teaching psychology? So first, what are the neurobiological consequences of arousal? And so, this is something you're all familiar with. It's a flight or flight response. We knew that the fight or flight response, as we typically talk about it, has two systems. It has a fast-acting sympathetic nervous system component. And here, this activates the adrenal medulla that releases norepinephrine and epinephrine. And so this is sort of underlying autonomic arousal. The skin conductance response you see, it's very fast-acting. You see a skin conductance response within a few seconds of something that's arousing. It can be very brief. It can go up and it can go down. And even very subtle, brief arousal results in this release of catecholamines, epinephrine, norepinephrine. The other part of of arousal we talk about is the hypothalamic pituitary adrenal axis, the HPA axis. This is slower, unfolds over several minutes, 20 minutes or so. It only comes out with more intense emotional experiences, so you can have a clear, subtle transient arousal response without having HPA axis activation, and this results in the release of cortisol. I'm going to focus on what I'm calling arousal here, which is this fast-acting sympathetic nervous system response. And so how does arousal influence explicit memory, the type of memory that depends on the hippocampus? Well, what I've put here is from a review article showing a connectivity map of the amygdala. So the amygdala, we think, really has two functions. One is the acquisition, storage, and expression of these simple threat associations. But it also, in the presence of arousing or emotional information, connects with several regions of the cortex to modulate processing in other cortical regions. And so the amygdala, if you look at this, it looks a little bit like Grand Central, in a map of the trains around New York, right? The amygdala has widespread connectivity in the brain. It modulates visual areas, it modulates cognitive areas, and it modulates memory areas when it detects something salient in the environment. And specifically, the amygdala modulates the hippocampus and parahippocampal regions, and it does this specifically through modulating the storage or consolidation of memory to enhance explicit memory retention. So I'm going to unpack that a little bit. Any of you remember who this is? Christine Blasey Ford, okay? A psychology professor testifying in Congress. And this is what she said. She talked about her memories of the assault from Justice Kavanaugh, and said just basic memory functions, and also just the level of norepinephrine and epinephrine in the brain that sort of, as you know, encodes that neurotransmitter that codes memory into the hippocampus. And so the trauma-related experience is locked there, whereas other details kind of drift. So she attributed this benefit in memory to norepinephrine, epinephrine, these catecholamines that are induced with arousal. And I know several memory researchers, including myself, that got calls from reporters saying, "Is she right?" And I was like, "Yeah, she's kind of right about how this works." And the data that she is right comes from the research of a gentleman named Jim McGaugh. So here's a slide I borrowed from a review article by Jim McGaugh, showing how this works. And so the way this works, and I'll talk about some of the data in a second, is that when you have arousal, you get the release of epinephrine, norepinephrine. That signals the amygdala when there's a learning event. The learning event initiates consolidation in the hippocampus and other brain regions, but I'm specifically interested in the hippocampus. But when there's an arousing event, the amygdala then modulates that consolidation process in the hippocampus. And damage to the amygdala impairs arousal-enhanced memory storage. And we know this is memory storage because research in rodents was able to administer epinephrine after an event and show the benefit on memory. And we know it's dependent on the amygdala, because if you damage the amygdala, you don't see this benefit, even though the rodent can still learn this. They just don't show the arousal enhancement of this learning. In fact, Jim McGaugh, and here I pictured, here's a picture of Jim, argues that, that one function of having consolidation, one function of this slow storage process that we have, why does it take time to store memories? He says one function is so that the emotional impact of an event that emerges after the event happens itself has the opportunity to modulate whether you remember that event. You want to remember events that are emotional, that are important, and by having this consolidation process that can be influenced by stress hormones, you are able to remember those events better. And so the question, of course, Christy Blasey Ford underwent a traumatic experience, but how much arousal is really needed to enhance memory? Because we want to do this in the classroom. And the answer is not much. So here's a study that we replicated. It was a study initially done in 1963 by two researchers named Kleinsmith and Kaplan. There were 60 Yale undergraduates. I did this early in my career when I was at Yale. They saw 8 word-digit pairs. And these were not particularly arousing words. They were like kiss or swim or chair. And we measured skin conductance while that people saw these word-digit pairs to the words themselves. And we just picked whichever words participants show a higher skin conductive response to as the higher arousal words, and whichever words they showed lower arousal response to as the low arousal words, and we divided them up that way. We then did a cued recall for the digits. What are the digits paired with the words? And we did this either immediately or after 24 hours. My collaborators on this were Mazar Banaji and Bob Crowder. And this is what we found. And so again, this is for the digits that were paired with the words, right? So here's the word, here's kiss, what digit was paired with it. And looking at percent recall. And what you see for the low arousal words is the most robust effect in all of memory, which is forgetting over time. But what you see for the digits paired with high arousal words is you actually don't show forgetting over time. In fact, you show a non-significant increase in memory over time. Now, emerging over time is consistent with enhancing consolidation. You see it over the retention interval. So that was not a lot of arousal. It was the arousal you have to the word like swim, for instance. We've done this now with several different types of stimuli. This here is the data I just presented to you. This is memories. These are pictures, highly emotional scenes. And what you can see in this case is immediately we see equivalent memory, but you showed a good retention for the arousing stimuli, not such good retention for the non-arousing stimuli. And so if I just looked immediately, I'd see 3 different effects of arousal on memory. But over time, what I see is enhanced retention of the emotional events. This particular paradigm I want to talk in a lot more detail because we did this in patients with amygdala damage to see if this is amygdala dependent in humans. And so this paradigm is something we call the taboo word paradigm. We wanted to induce some arousal, so instead of relying on words like swim that might randomly induce arousal in some individuals and not others, we use dirty words, the kind of words you wouldn't expect to here in the laboratory. So, the neutral words were things like chair or keep. The dirty words were things like, I'm not doing that, sorry. Okay. We had 22 amygdala lesion patients and 22 healthy controls. They had 20 taboo words and 20 neutral words. We measured the skin conductance responses to the words themselves, and we did verbal arousal ratings as well. And we looked at recall immediately and after an hour delay. And I should say the work by Kleinsmith and Kaplan showed that the crossover point for this learning is about 20 minutes. So by an hour, you start to see these effects. And so this is what we see. So as you can see, the control participants showed some forgetting over time. We had multiple recalls, so it's not a lot of forgetting. Whereas they showed flat recall or somewhat improvement over time for the arousing stimuli. The patients still showed a benefit for the taboo words versus neutral words. So there's several things about emotion that can enhance memory. It was a category of emotion words, right? So the semantic similarity, whereas it was sort of random and neutral words. So that can enhance memory. That's fine in these patients, right? But what they don't show is enhanced retention of the emotional words over time. And so, this showed that sort of an event that occurs at the time of encoding, sort of arousal that occurs at the time of encoding can improve memory. But one thing you might want to ask is, that's great. Like, I can introduce something that's exciting in my classroom. I can kind of get people a little excited. They'll remember that better. But what about the rest of it? Not everything does that, right? Well, here I want to talk about, when in the memory cycle arousal can enhance memory. And I'm going to talk about the memory penumbra. This is sort of a word that we've started to use, which not many people are familiar with. I wasn't familiar with it when we first started to use it. But here's a couple definitions. So the penumbra from the dictionary is a partially shaded outer region of the shadow cast by an opaque object. It's something around an object. In law, it's the group of rights derived by implication of other rights explicitly protected. So it's things that are around the event. And so I'm going to talk about the penumbra of memory. When does arousal actually enhance memory? We can think about memory sort of as a time-dependent process. You learn something, you encode the memory, the time goes by, your memory is consolidated, you retrieve the memory. What I've suggested to you so far is arousal at encoding can enhance memory. There's also research, I'm not going to talk about in detail, saying arousal prior to learning can carry over and enhance memory. And we see this in both behavior and in the brain. So the sort of pattern of brains, we see amygdala activity. If somebody's in an aroused state, it will carry over and enhance memory for information encoded at that time when arousal is still high. And also, arousal after learning can enhance memory for related material. It doesn't enhance memory for all material, but for related material. So what we know is there's several time windows. So essentially, arousal enhances memory for things that occur around the time of the arousing event. We do know, however, that arousal can impair memory as well. So first, very prolonged or extreme arousal impairs memory at all stages. And arousal and stress prior to retrieval can impair memory. So retrieval requires, the prefrontal cortex organizing your thoughts. Even mild arousal can impair some of those functions. So sort of arousal prior to retrieval, particularly stronger arousal can impair memory, not so much mild arousal. So just to talk about now, how does this inform the teaching of psychology? I've suggested that inducing arousal in the classroom can enhance retention of class materials. And based on the principles of the neurobiology of memory arousal, we know that even subtle shifts, transient shifts in arousal, such as surprising information, engaging or entertaining lectures, can result in catecholamine release, just anytime you see that skin conductance response. And this can enhance memory retention. That arousal can enhance memory for materials learned before or after arousing events. This is the penumbra of memory. That the effects of arousal memory will be most pronounced after delay, so it affects consolidation. That takes time, retention, at least an hour. That extreme or prolonged stress will impair memory. So, many of you may have experienced your students not performing as well during the COVID pandemic. Everybody was stressed, including the teachers, but students, I think, were hit particularly hard. And then arousal or stress prior to testing can impair retrieval and test performance. So this is, anything you can do to help your students stay calm in the moment of a test will help performance. So just to summarize this section of the talk, I've suggested although there are many reasons to aim for entertaining and engaging presentations in the classroom, one specific benefit is that the subtle shifts in arousal will enhance the retention of classroom materials. So I started this talk saying, one of our goals in psychology is to create a learning environment that enhances the retention of insights from psychological science. I've tried to present to you some of the research that I've conducted and others in my field that have helped me inform this goal in my classrooms. And I suggested that enhanced memory retention, you can enhance memory retention through inducing reconsolidation, that delayed and incomplete retrieval practice that can enhance memory by restoring the memory, strengthening the memory, and that you can enhance memory retention with mild arousal. Even subtle shifts of arousal in the classroom can lead to better memory. So with that, I want to thank you for your time and attention. And I'm happy to take any additional questions if we have time. Do we have time?
Thank you. That was a great talk. I was wondering if your model, the first half, the consolidation model, does that apply also for implicit memories? We talk mostly about explicit memories, but does the same thing?
Well, I'm talking about explicit memory because that's the one we care about. But we could call threat-related memories implicit, and that they're implicitly expressed. It's also been shown in motor memories.
Oh, that's what I was thinking about, more procedural memories, because as you were talking, I started taking piano lessons as an adult, and so I drive my piano teacher crazy about this because I was thinking about this a lot in terms of how we could pair this with sleep, so what we heard in the teaching slam yesterday. So a lot of what I've noticed is when you practice music and then you sleep on it, so a lot of times it's really frustrating, but if you sleep on it, those memories seem to consolidate overnight, and you can actually see that change overnight in terms of practice. So I was starting to think about if you can disrupt those memories in your practice. A lot of times, like I talk with my teacher about strategies of how to try to activate those memories in terms of consolidation. Like, don't just start practicing in the same spot all the time. Try to activate them in different ways, and then you can try to strengthen that memory using the kinds of things you were talking about.
Exactly. I mean, the thing about reconsolidation, it's a natural memory process that's been around for a long time, right? I mean, forever. It happens, it's what our memory does. We haven't identified exactly when and how it occurs until recently, right? So all of those phenomena that you're talking are very consistent with the idea that, starting in a different place, right, will induce reconsolidation. Sleep, we know, is important for consolidation period, whether it's consolidation or reconsolidation. It's thought to underlie the dynamic nature of memory. And we've known memory is dynamic for a long time. We just didn't know exactly how and why. But now that we do, we can start to think about more specific ways we might target it.
So when it's frustrating, just sleep on it.
Make my way over to a question over here. But one of the things I'm also realizing is that shift in attention that's important. So I should resist the urge to be constantly entertaining in my class. So
I would like to come personally, but
Oh, thank you. Oh
my gosh. I don't know if that's me.
That was me. Sorry about that. Yes.
That's one way, I suppose. Sure. So forgive me, I don't have the neural background, but I'm thinking about the ways to get those subtle arousals, and I'm very interested by your study with the taboo words. And so I'm thinking is like a shared laugh, or is just saying something that you want your students to remember, like, oh shit, this might be important. And that, is that enough? Or what kind of ways would you actually recommend in the classroom?
This is the hard question, right? I mean, in that study by Kleinsmith and Clapton I talked about, some people found the word swim more arousing than the word kiss, and other people found the word kiss more arousing than the word swim, right? So, in the taboo word study, we just really hit them over their head by giving them very dirty words. So I think it's hard to know for one individual, because I think it depends on their interpretation somewhat what they're going to find arousing. We can think of general principles, things that are funny, things that are particularly interesting, for somebody, but that might not be interesting for somebody else. So here, I think the collective wisdom in this room is going to be much better than what I can tell you in terms of the kinds of things that get students engaged. But I will say, pupil dilation is also a measure of the locus coeruleus, which is involved in the release of norepinephrine. And we see pupil dilation when people are working hard, right? Sometimes, sort of effort type things. So it's very subtle. You see it constantly. So people, things that are surprising, prediction errors, right, induce pupil dilation. So, but again, I think it somewhat depends on the individual and their interpretation, because it is sort of inherently an emotional response that's dependent on their appraisal. But, some things are more universal than others, but I think everybody here in this room could probably give you insights into what works for them. I wouldn't suggest saying curse words, though.
We have time for one last question.
Hi. I had, I guess, two-part question. If you could briefly repeat the safety condition versus the extinction condition.
They're identical.
Oh.
So I called it safety because that's really what you think you're learning in that circumstance, like safety learning. And extinction implies, that it's sort of a process that is engaging a new memory. So, but the procedure is identical.
And is the reason that you think that the second memory trace, second, third, fourth memory trace are progressively taking up more competition or giving more competition to that first memory, and that's why the first memory begins to not fade but have less of an impact?
So you're talking about the reconsolidation, or you're talking about no? So the notion is during reconsolidation, what happens is the memory's active. And when it's active, it's going to be restored. And so you could restore the memory with new information available at the time of retrieval. So the memory is transformed through this process, right? Okay. It's not that, if no new information comes in, then the memory just gets restored and it gets strengthened. Okay. But if new information comes in, then it's updated to include that information.
Thank you.
Thank you so much. Let's take that. Lunch in the pavilion, and
All right. Hi.